Hi everybody. This set of lectures is going to be on the topic of translation of the mRNA molecule into a protein. So in the previous video, I've covered prokaryotic translation initiation. I just want to repeat the important points about the process. The three important points is that the small ribosomal subunit binds first, and it binds together with the starting tRNA already preloaded into the ribosomal subunit, while the E and the A side of the small ribosomal subunit are occupied by initiation factors to prevent tRNAs from binding there. Another important point is that prokaryotic ribosomes need to have a Shine-Dalgarno sequence in front of the starting AUG in order to properly bind in that location and properly set the frame of translation. Part of the reason why prokaryotes require a particular sequence for translation initiation is because prokaryotes tend to have multiple proteins on one mRNA. They like polycistronic messages. So there will be different ribosomal entry sites along the mRNA. So, and then the third important event that happens is that GTP that was brought by the various initiation factors that bind the small subunit is hydrolyzed, and that causes them to let go of the small ribosomal subunit, the 30S subunit, and that the combination of the 30S ribosomal subunit plus the tRNA in that location now will allow the 50S subunit to bind, assembling the ribosome that they can now proceed to translate the message. So if you look at the next slide, this shows the eukaryotic translation initiation. And if you look at it at first, it seems overwhelming because there's a lot more blue blobs with names. But the process is actually simpler than prokaryotic because let's remember the three important points. The fact that the 40S ribosomal subunit, the small subunit, will bind first is correct also for eukaryotic translation initiation. It will also bind first, and it will also have, right here, the starting tRNA loaded up already. The second point of prokaryotic ribosomal translation was that it required a special sequence in order for the ribosome to bind, the Shine-Dalgarno sequence. Unlike the prokaryotic translation, the eukaryotes don't have a special sequence. They have a Kozak sequence and it kind of helps things along, but really what is recognized by the initiation factor 4F is the 5' cap of the mRNA, because unlike prokaryotes, eukaryotes usually pack only one message, overwhelmingly, only one message per mRNA. There's only one protein per mRNA is translated from in eukaryotes. And then the third step is, again, GTP is GTP is hydrolyzed to GDP. That causes the various initiation factors to release and let go, and the 60S subunit recognizes this whole package of a tRNA and a 40S, assembling the complete ribosome. So here are the steps. Before we go through this just a little bit more detail, I just wanted to take away the fear of the acronyms. E, there you can see that that overwhelmingly all the little blue blobs on the diagram are called EIF something. E stands for eukaryotic, I stands for initiation, and F stands for factor. And they're just numbers afterwards, depending on when and how they were identified when people worked on translation initiation back in the 70s. Okay, so the initiation factor 1, 3, and 1A are the initiation factors always associated with the 40S subunit whenever it's not attached to mRNA. And they keep the E side, the F1 keeps the E side, and 1A keeps the A side occupied so that tRNAs cannot bind there. Or, the elongation factors, lots of things can potentially get in there. And then the IF2 and IF5B are two GTP bound proteins that will bind to the small 40S subunit and deliver the first tRNA into it, which will be placed into the P side of the ribosome. You can see that they're both carrying GTP because while they're very good at in their conformations when they have GTP attached to them, they easily bind into the areas they're supposed to bind. So the IF binds to the 5B binds to 1A, placing the tRNA with methionine just so into the P side. And where's the other guy? And EF2A leaves. Oh no, it's right here. They just forgot it in the diagram. The EF2 is also bound to the methionine. And then this whole assembly, now that it has a tRNA loaded into it, it will be able to both recognize the 4F initiation factor that's bound to the 5' cap of the mRNA, and then this whole arrangement will proceed to scan along the mRNA, looking for the first methionine codon. So there's no Scheindel-Gardner sequence in eukaryotes. It literally is the first methionine. Whatever they find first will establish the frame of translation. So once the tRNA that was methionine has properly base paired with the starting AUG codon, a big event occurs. Because this whole assembly is stuck here, the GTP on F2 and GTP on F5B are hydrolyzed. That changes their ability to bind to the ribosome, and all of the factors that were keeping the sites occupied, preventing translation initiation, come off when GTP is hydrolyzed to GDP, leaving just the starting methionine tRNA bound to the mRNA, bound to the small ribosomal subunit. And that is the arrangement that the large ribosomal subunit, which is 60S in eukaryotes, can now recognize. So, step 1, have a fully loaded small ribosomal subunit with the methionine tRNA bound in it. Step 2, the initiation factor 4F has recognized the 5' cap and is sitting on the mRNA to be translated. Step 3, everything assembles. The small ribosomal subunit plus the tRNA, plus all of their little helpers, bind the initiation factor 4F that's attached to the 5' cap. Step 4, 5' cap here. Step 4, scan. Here go. Scan along the mRNA. You can see that the mRNA is bent and folded, but the initiation factor 4F is attached both to the 40S ribosome and to the cap. It doesn't come off of it. So as soon as the first methionine is encountered, the three base pairings are formed between the tRNA and the starting methionine codon. All of the little helpers come off because GTP has been hydrolyzed to GDP, and they no longer have the right conformation to remain attached to the small ribosomal subunit. That leaves an empty ribosomal subunit, small ribosomal subunit to which the large ribosomal subunit with the catalytic domain in it can bind. So very important, GTP has to be hydrolyzed in order for the initiation factors to let go of the ribosome, of the small ribosomal subunit. And as I've said before, and I'm going to repeat, there is no Scheindel-Gardner, there's no particular sequence in front of the starting AUG codon in eukaryotes. There is a very vague Kozak sequence. The AUG bolted here is the starting methionine codon. It helps the ribosome it helps translation initiation if the fourth nucleotide, the nucleotide right after the AUG is a G. That does help translation rates. But many proteins that we have do not have any sort of recognizable Kozak sequence. Many of them don't have this second G, so the Kozak sequence, unlike the Scheindel-Garner sequence in prokaryotes, which is required, the Kozak sequence in eukaryotes is a gentle suggestion. So, as I said to you guys right now, that eukaryotes kind of stick to this one mRNA, one protein rule. So, is it impossible for in eukaryotic cells to have multiple places for translation initiation, aka internal ribosomal entry sites, also frequently just abbreviated as IRIS? Well, you have to. There's a certain kind of organisms that love to prey on eukaryotic cells, and those are viruses. They have tiny genomes. They can't afford one mRNA, one protein situation. They basically have one mRNA. So a lot of the viruses have come up with ways to trick the eukaryotic translation initiation machinery and have created these irises. So all of these examples are either viral viruses getting around the eukaryotic one mRNA, one protein rule, or there are certain situations in eukaryotes when multiple proteins can be transcribed from one message, but they're very rare. So there's two ways that this question can be approached by the irises. You can either you can have irises that look like the 5' cap for the initiation factor 4F. So instead of just binding the 5' cap, the iris, initiation factor 4F also recognizes the iris, binds there, and that allows for the small ribosomal subunit to bind in that location as well. There's other solutions where instead of bothering to recruit the initiation factor 4F to the location where they would like the protein to be translated from, you can have a structure that looks like not the 5' cap, but the actual initiation factor 4F. So this also allows the ribosome, just the 440S subunit, to bind in that location and triggers initiation of translation in that location. So you can have multiple, if you have irises, internal ribosomal entry site, a ribosome can also enter an iris and then start translating the following message after that. The starting AUG the first available AUG will still set the frame of translation. That's how eukaryotes do it. So one important thing to realize is that irises will never be as efficient as a proper 5' cap. They're always the fakes that can get the job done, but are not the ones that will have high translation efficiencies, will not attract the most ribosomes. Another thing that is not mentioned often when you talk about translation, I mean, you guys all know that ribosomes translate and so forth, is that frequently textbooks will draw the mRNA molecule as a line. There's a distinct beginning and a distinct end. All of these diagrams really kind of make it seem like it's just a string and the ribosomes kind of roll along of it. However, in eukaryotes, the mRNA is always circularized. That's just one of the ways to protect the mRNA from the RNases that are lurking in the cytoplasm, ready to chew up any bad RNA that they can encounter, which is usually virus. So when initiation factor 4F binds, it also recruits two other factors, 4E and 4G, which will then are capable of binding to the poly-A binding proteins that coat the poly-A tail when the mRNA exits the nucleus. This causes a circular mRNA molecule, and as soon as the ribosomes fall off here, when they encounter the stop codon, they're actually really close to rebind again to the at the 5' cap area and continue translation. whenever this binding between 4G and the poly-A binding proteins is abolished for one reason or another, and the message is linearized as opposed to being circular here, it becomes a lot more fragile, and it degrades much faster by the RNases that are just looking forward. They're just trying to find a way to get there and degrade the RNA message. Another important thing is that just like in prokaryotes, you have polysomes, so a bunch of ribosomes sitting on the RNA message and processively translating it. Same thing occurs in eukaryotes as well. Because the message is circular, you can just keep on going. The same ribosome, as soon as it's done translating, is in very close proximity to the 4F cap and can rebind and restart translation.